And we are back with another chapter of advanced pathophysiology. Today we're going to cover chapter 15, the structure and function of the neurologic system. We'll be reading the entirety of this chapter, so even though my pages may not line up perfectly with yours, we'll be covering all of it. If there's sections that I'm going too quickly over or anything like that, always feel free to pause. So, without much further preamble, let's jump right into this chapter. The nervous system is a remarkable structure responsible for body's ability both to interact with the environment and to regulate and control activities involving internal organs, muscles, and glands. It is a network composed of complex structures that transmit electrical and chemical signals between the brain and the body's many organs and tissues. So, an overview and organization of the nervous system. Although the components of the nervous system are considered individually to facilitate a better understanding of their function, it is important to recognize that these distinct parts of the nervous system function as a unified whole. Most broadly, the nervous system is divided into the central nervous system and the peripheral nervous system. The central nervous system consists of the brain, spinal cord, enclosed within a protective cranial vault and vertebra, respectively. The peripheral nervous system is composed of cranial nerves, which project from the brain and pass through foramina, or openings in the skull, and the spinal nerves, which project from the spinal cord and pass through intervertebral foramina. Your peripheral nervous system, or PNS, from the spinal cord pathways can be divided into ascending or afferent pathways that carry sensory information toward the central nervous system, or descending or efferent pathways that innervate effector organs, such as your skeletal, cardiac, and smooth muscle, as well as some of those glands. Most peripheral nerves carry a combination of both afferent and efferent pathways. Cranial nerves are viewed most correctly as modified spinal nerves. Cranial nerves control motor and sensory function, similarly to spinal nerves, but also can have specialized sensory tasks, such as smell, taste, sight, and hearing. Functionally, the peripheral nervous system can be divided into the somatic nervous system and the autonomic nervous system. The somatic nervous system consists of motor and sensory pathways regulating voluntary motor control of skeletal muscle. The autonomic nervous system, or ANS, also consists of motor and sensory components and is involved with regulation of the body's internal environment, your viscera, throughout involuntary control of organ systems. The ANS is further divided into sympathetic and parasympathetic divisions. Although most aspects of the ANS are involuntary, some aspects can be controlled through mental practice, with or without the biofeedback techniques. So, cells of the nervous system. Two basic types of cells comprise nervous tissue: neurons and the supporting neuroglia. The neuron is an electrically excitable cell and transmits electrical or chemical information between the neurons or to the effector organ. Neurological cells provide structural support, protection, and nutrition for the neurons and facilitate neurotransmission. Neuroglia cells include astrocytes, microglia, and oligodendrocytes in the CNS, and Schwann, neurolemma, and satellite cells in the peripheral nervous system. So, talking about neurons specifically here. Neurons. Working alone or in units, neurons detect environmental changes and initiate body response to maintain a dynamic steady state. Neuronal structures vary markedly, so that each neuron is adapted to perform specialized functions. Although they may vary somewhat in function, neurons have three common components. They have a cell body, dendrites, and axons. Cell bodies for most neurons, even those extending axons into the peripheral nerves, are located within the CNS. Dense collections of cell bodies in the CNS are called nuclei. In the peripheral nervous system, or PNS, cell bodies can be found in groups called ganglia, or plexuses. The dendrites are extensions that carry nerve impulses toward the cell body. The dendritic zone is the receptive portion of the neuron that receives the stimulus and either propagates or diminishes conduction. Axons are long projections that carry nerve impulses away from the cell body. The axon hillock is a cone-shaped process where the axon leaves the cell body. The first part of the axon hillock has the lowest threshold for stimulation, so action potential often begins there. The principle of divergence refers to the ability of axonal branches to influence many different neurons. Convergence applies when branches of various number of neurons converge on and influence a single neuron. So here we have a picture of that neuron breakdown. So I'll leave that up for just a moment. Again, you can always pause to take a look at things longer if you feel like you need it, but I'll keep reading on the side, okay? The cellular constituents of a typical neuron include microtubules, neurofibrils, microfilaments, and nicyl substance bodies that are involved in protein synthesis, although most neurons are non-dividing cells. Some neurons continue to divide after birth. For example, the olfactory neurons in the nose continue to divide throughout life. A typical neuron has only one axon, which may be tightly wrapped with a segmental layer of lipid material called myelin, an insulating substance. In the brain and spinal cord, myelin is formed by oligodendrocytes. Regions of the brain and spinal cord with a high level of myelination constitute the white matter, whereas regions lacking significant myelination are gray matter. In the PNS, the myelin sheath of motor and sensory axons is formed by Schwann cells. The myelin sheaths are interrupted at regular intervals by the nodes of Ranvier. Axons can branch at the nodes of Ranvier. Nutrient exchange is not possible through the myelin sheath, although it can occur at the nodes of Ranvier, where the axon is not insulated. Myelin acts as an insulator that allows an action potential to leap between the nodes of Ranvier rather than flow along the entire length of the membrane, yielding increased velocity of conduction. This mechanism is referred to as the saltatory conduction. Disorders of myelin sheath, a demyelinating disease, such as multiple sclerosis, Guillain-Barré, Charcot-Marie-Tooth, demonstrate the important role of myelin that plays in the nerve conduction. Conduction velocities depend not only on the myelin coating, but also on the diameter of the axon. Larger axons transmit impulses at a much faster rate. Neurons are structurally classified on the basis of the number of processes, or projections, extending from the cell body. There are four basic types of cell configuration. The first, unipopular. Second, pseudo-unipopular. Third is bipolar. And fourth is multipolar. So, talking about the first, unipopular neurons have one process that branches shortly after leaving the cell body. For instance, those found in the retina. Pseudo-unipopular neurons also only have one process. The dendritic portion of each of these neurons extends away from the CNS, and the axon portion projects into the CNS. The configuration of a typical of sensory neurons is both cranial and spinal nerves. Your bipolar neurons have two distinct processes arising from the cell body. This type of neuron connects to the rod and cone cell of the retina, for instance. And then your multipolar neurons are the most common and have multiple processes capable of extensive branching. A motor neuron is typically multipolar. So here we're taking a look at that different type of multipolar, bipolar, pseudo-unipolar neurons. And I'll leave this up for just a moment, and I'll keep reading. So functionally, there are three types of neurons. The first is sensory, the second is associational, and the third is motor. Sensory neurons carry impulses from the peripheral sensory receptors to the CNS. Associated neurons, or interneurons, transmit impulses from the neuron to neuron and are located solely within the CNS. Your motor neurons transmit impulses away from the CNS to an effector organ, the skeletal muscle of organs. In skeletal muscle, the end process of the axon forms specialized structure called neuromuscular junction. So here are major types of sensory receptors. I'll leave that box up for just a moment. You can read through those so that you have some idea, but we'll keep on going, okay? Neuroglia and Schwann cells. Neuroglia, the nerve glue, are the general classification of non-neuronal cells that support the neurons of the CNS. Your conduction velocities depend not only on the myelin coating and nodes of Ranvier, but also on the diameter of the axon. Larger axons transmit impulses at a faster rate. Your oligodendroglia form myelin sheaths within the brain and spinal cord. Regions of the brain and spinal cord with a high level of myelination constitute the white matter, whereas regions lacking significant myelination are gray matter. Schwann cells form the myelin sheath around axons in the peripheral nervous system and direct axonal regrowth and functional recovery of injured neurons. Your non-myelinating Schwann cells provide metabolic support. Characteristics of neuralgia and Schwann cells are summarized here in this figure. So here I'll leave the types of neuralgia cells up. Again, feel free to pause if you need anything specific or if you want to read that section specifically. Otherwise, I keep going. Table 15.1, the support cells, your glial cells of the nervous system. Again, these boxes, pause if you need to read anything specific. But quick overview, astrocytes. The oligodendroglia, your Schwann cells, your microglia, and endomyel cells. Ependymyal cells. Woo. And then there is a little report. I'll leave this up for just a moment. Pause if you want to read this yourself. I'll leave that up for just a second. So again, you can stop and pause. But otherwise, I keep going. Nerve injury and regeneration. Mature neurons do not divide. An injury in the CNS causes permanent loss of damaged neurons. Peripheral nerves can repair themselves through local, anterograde, and retrograde changes. This is known as axonal reaction. Local changes occur when the axon is severed. The cut ends retract and the axolemma covers the cut ends, diminishing the escape of axoplasm. Macrophages and Schwann cells begin to phagocytize damaged tissue. The cell body undergoes chromatolysis, with swelling, loss of nicetal bodies, and lateral migration of the nucleus. Your antegrade degeneration occurs in the distal axon. A characteristic swelling appears in the axon terminal, and it degenerates and loses contact with the postsynaptic membrane within about 7 days. Macrophages and Schwann cells phagocytize the remnants of the axon terminal, and Schwann cells proliferate, forming a column or tube of Schwann cells enclosed by the original basal lamina of the endoneuronium. A retrograde change occurs at the proximal end of the injured axon and are similar to antegrade changes, but only back to the next node of Ranvier. Approximately 7 to 14 days after the injury, a new terminal sprouts, project from the proximal segment, guided by Schwann cells, and enter the sustaining substrate of the Schwann cell column, or tubes, for axonal regrowth. So, this following figure contains a more detailed representation of these events. This process is very slow, only about 1 millimeter per day, and is limited to myelinated fibers in the peripheral nervous system. The regeneration of axonal constitutes in the CNS is limited by an increased incidence of glial scar formation and the different nature of the myelin formed by the oligodendrocyte. Feel like they could have added that picture to that page, but we'll stop here so you can take a look at it. So here again, we have a normal neuron with an injury, and that neuron is connected to normal muscle skeletal tissue. So then 2 weeks after injury, you see how it breaks down via macrophages and Schwann cells. Then 3 weeks after injury, and then about 3 months after that injury, there is the reformation. Whereas there is an unsuccessful nerve regeneration and it snaps and it closes off. Which leads to the question, should we then try and cut that piece off, let it try and fix itself again, but try and let it fix to the new portion? Questions to ask a neurologist, or neurosurgeon rather. Your nerve regeneration depends on many factors, such as the location of the injury and the type of injury, the presence of inflammatory response, and the process of scarring. The closer the injury is to the cell body of the nerve, the greater the chance that that nerve cell will die and not regenerate. A crushing injury allows recovery more fully than does a cut injury. Crushed nerves sometimes recover fully, whereas a cut nerve often form connective tissue scars that block or slowly regenerating axonal branches. The peripheral nerve injury close to the spinal cord recover poorly and slowly because of the long distance between the cell body and the peripheral termination of the axon. So that answers, I guess, that question. Your nerve impulse. Neurons generate and conduct electrical impulses by selectively changing the conductance across their plasma membrane and influencing other nearby neurons by the release of chemical signals, those neurotransmitters. An unexcited neuron maintains a resting membrane potential. When the membrane potential is raised sufficiently, an action potential is generated and propagated to other parts of the neuron. The action potential occurs only when the stimulus is strong enough. If it is too weak, the membrane remains unexcited. The property is sometimes called an all or none response. Your synapses. Neurons are not typically continuous with one another, but form points of contact with adjacent neurons with a specialized structure called a synapse. The synapse is composed of a small bulbous end of the presynaptic neuron, a synaptic knob, that is separated from the postsynaptic neuron by a synaptic cleft. Impulses are transmitted across the synapse via chemical and electrical conduction. Only chemical conduction is discussed here. So chapter 1 contains information on electrical conduction. The neurons that conduct a nerve impulse are named according to whether they relay impulses toward presynaptic neurons or away from the synapse, postsynaptic neurons. Again, four basic types of connections occur in the region of contact between the presynaptic and postsynaptic neurons. These are between axons, from axon to cell body, from axon to dendrite, and from dendrite to dendrite. In response to the arrival of an action potential at the synaptic knob, vesicles containing neurotransmitters release their content into the synaptic cleft. Neurotransmitters diffuse across that synaptic cleft and bind to specific receptors on the postsynaptic neurons, where they trigger an action potential in the postsynaptic neuron. Brain synapses can change in strength and number throughout life. This is known as a synaptic plasticity or neuroplasticity. So here you see the neuronal transmission of the synaptic cleft. Again, pause if you want to look at more specific sections of this. Otherwise, I keep reading. Neurotransmitters. Neurotransmitters are synthesized in the neurons and localized in the presynaptic terminal, a synaptic bouton. Neurotransmitters are released in the synaptic cleft in response to the arrival of an electrical impulse and bind to a receptor site on the postsynaptic membrane of an adjacent neuron or other effector. Here, binding of the neurotransmitters to its receptor causes a change in conductance of postsynaptic membrane, allowing propagation of the impulse. Efficient termination of the signal requires rapid degeneration of the neurotransmitters in the cleft on the postsynaptic cell surface. Neurons can synthesize more than one neurotransmitter, and postsynaptic membranes can contain more than one type of transmitter-specific receptor. Common neurotransmitters include norepinephrine, acetylcholine, dopamine, histamine, and serotonin. These have been varied and overlapping functions that are summarized here on this table. So again, pause. All of these are going to be important to know at one point or another, especially on like how they affect the body and its clinical examples. Your acetylcholine, your norepinephrine, your serotonin, dopamine, histamine, your amino acids, your gamma aminobutyric acid, your GABA, your glycine, your glutamate, and aspartate, your endorphins and enkephalins, your substance P, vasoactive intestinal peptide. So I kind of went over like this general section quickly. Pause, pause, pause. I'm going to go back up so you can read sections, but please pause, read these sections. It's always good to know this stuff. Especially if you don't have the book and you can't look it up at your own chapters. Hit the pause button, read it, write it down, make some flashcards if it helps you memorize how they work. It never hurts to have this. So, because the neurotransmitters is normally stored on the presynaptic side of the synaptic cleft, and the receptor sites are on the postsynaptic side, chemical synapses operate in one direction. Therefore, action potentials are transmitted along the multineuronal pathway in one direction. The binding of neurotransmitters at the receptor site changes the permeability of the postsynaptic membrane and, consequentially, its membrane potential. Two possible scenarios can follow. The postsynaptic neuron may be excited, so it's depolarized. Your excitatory postsynaptic potentials, or the postsynaptic membrane may be inhibited or hyperpolarized. Inhibitory postsynaptic potential. Again, Chapter 1 contains the review of electrical impulses and membrane potentials. I may repost that chapter. I've read it, I just never posted it, so I may have to reread it because I didn't record it. All depends on if I can get to it or not. Usually, a single postsynaptic potential cannot induce a neuron's action potential and the propagation of the nerve impulse. Whether an action potential occurs depends on the number of and frequency of excitatory and inhibitory potentials. The postsynaptic neurons receive a concept known as summation. Your temporal summation refers to the effects of successive, rapid impulses received from a single neuron on the same synapse. So again, temporal, single, same synapse. Your spatial summation is the combined effect of the impulses from a number of neurons on a single synapse at the same time. So that's multiple neurons on a single synapse. Your facilitation refers to the effect of multiple EPSPs on the plasma membrane potential. The plasma membrane is facilitated when summation brings the membrane closer to the threshold potential and decreases the stimulation required to induce an action potential. The effect of a neurotransmitter on the membrane potential depends on the balance of these effects. The mechanisms of convergence, divergence, summation, and facilitation allow for the integrative processes of the nervous system. 2 points could be helpful in the understanding the complexity of the brain physiology. First, the aforementioned neuromodulators appear to function to raise or lower the membrane potentials of neurons. These chemicals facilitate or inhibit the effect of neurotransmitters. The second, reciprocal synapses between dendrites, that is, one dendrite being able to depolarize or hyperpolarize the membrane potential of another dendrite through the use of neurotransmitters, demonstrate that the interactions between neurons are far more complicated than postulated by simple on-off models of brain function. So talking about the big picture here, the central nervous system. The central nervous system is composed of the brain and spinal cord. The neuron is the basic structure of the CNS. So let's talk strictly brain. The brain is a functionally integrated circuit of millions of neurons with different structures, molecular composition, networks, and connections. It's roughly only 3 pounds, but receives 15 to 20% of the total cardiac output. The brain enables a person to reason, function intellectually, express personality and mood, and perceive and interact with the environment. The three major structural divisions of the brain are the forebrain, or prosencephalon, which includes the telencephalon and diencephalon. Ooh. The midbrain, the mesencephalon, which connects the pons to the diencephalon and include the corpora quadrigemina, the tegmentum and cerebral peduncle, and the hindbrain. So let's just call it the forebrain, the midbrain, and the hindbrain, which include the cerebellum, the pons, and the medulla in the hindbrain. The midbrain, medulla, and pons compromise the brainstem, which connects the hemisphere of the brain, cerebellum, and spinal cord. A collection of nuclei within the brainstem collectively constitute the reticular formation. The reticular formation is a large network of diffuse nuclei that connect the brainstem to the cortex and control vital reflexes, such as cardiovascular function and respiration. It's essential for maintaining wakefulness and attention, and therefore is referred to the reticular activating system. So here we have the divisions of central nervous systems, your forebrain, midbrain, and hindbrain, and your spinal cord, and its secondary vesicles and associated structures. Again, pause if you want to read the specifics. Here is a picture of the physicality of your forebrain, midbrain, and hindbrain. And here is the reticular activating system. Again, pause if you want to read that, study it, look at it a little closer. Divisions of the brain are associated with specific functions, but it is important to recognize that most brain functions require integration of multiple different inputs from different parts of the brain. However, for clinical considerations, functional specificity is useful for localizing pathologic conditions in various nervous system regions. Corbinian Brodmann, a German neuroanatomist, was among the first to define structural and functional aspects of brain anatomy. Brodmann's area are still used to correlate functional activities to many regions of the cerebral cortex. Figure 15.8c illustrates these regions and describes some of the areas. The mapping of brain networks using functional MRI and other advanced imaging techniques is helping us discover new insights into the function of these complex and interconnected pathways. So again, here we have different pictures of what to expect in the brain. Again, I'm gonna let you pause here, read them, review them, just so you have a general idea of where is what. And here is box 15.2, the brain network. Again, read this at your own pace. Pause if you need to. The forebrain, also known as the telencephalon, consists of the cerebral cortex and the basal ganglia. The surface of the cerebral cortex is covered with convolutions called gyri that greatly increase the cortical surface area and the number of neurons. These grooves between adjacent gyri are termed sulci. Deeper grooves are called fissures. The cerebral cortex contains an outer layer of cell bodies of neurons, your gray matter. Gray matter is organized into columns perpendicular to the surface that receives, integrates, stores, and transmits information. Your white matter lies beneath the cerebral cortex and is composed of myelinated nerve fibers, again, those axons. The two cerebral hemispheres are separated by a deep groove known as the longitudinal fissure. The surface of each hemisphere is divided into lobes named after the region of the skull under which each lobe lies. The posterior margin of the frontal lobe is the central sulcus, the fissure of Rolando, and it borders inferiorly on the lateral sulcus, the Sylvian fissure, or lateral fissure. The prefrontal area is responsible for goal-oriented behavior, your ability to concentrate, short-term or recall memory, and the elaboration of thought and inhibition of on the limbic system, your emotional areas of the CNS. The premotor area, the Brodmann area 6, is involved in programming motor movement. This area also contains the cell bodies that form part of the basal ganglia system, your extrapyramidal system afferent pathway. Outside the pyramids of the medulla oblongata, the frontal eye fields, which are involved in controlling eye movement, are located in the middle front gyrus. The primary motor area, Brodmann area 4, is located along the precentral gyrus, forming the primary voluntary motor area, which has a somatotopic organization that often is referred to as the homunculus, or little man. Electrical stimulation of specific areas of this cortex can cause specific muscles of the body to move. For example, stimulation of the Brodmann area 4 in the medial longitudinal fissure affect the lower limbs of the foot. where stimulation of the superior lateral surface of the precentral gyrus affect the torso and arm, the middle third of the hand, and the lower third of the face and mouth and throat. The axons traveling from the cell bodies in and on either side of the gyrus project fibers, or axons, that form the pyramidal system. The system includes the corticobulbar tract, that's synapses in the brainstem, and provides voluntary control of muscles in the head and neck, and the corticospinal pyramidal tracts that descend into the spinal cord and provide voluntary control of muscles through the body. Your cerebral impulses control function on the opposite side of the body, a phenomenon called contralateral control. The Broca area in the inferior frontal lobe, that's Brodmann areas 44 and 45, is an important center for speech and language processing. This area, rostral to the inferior edge of the premotor area, that's Brodmann area 6, on the inferior frontal gyrus, is usually most important in the left hemisphere. Injury to that area results in difficulty forming or inability to form words. Your expressive aphasia and dysphagia, which is going to be discussed in chapter 17. So, again, knowing different Brodmann areas can help you, for instance, figure out where certain strokes are occurring in a patient. So, here we're taking a look at your primary somatic, motor, and sensory areas of the cortex. I'll leave that up so you can have a quick peek and view of what we're looking at. And again, examples of that somatic, motor, and sensory pathway. I'll leave that up for just a moment. Again, pause, pause, pause. The parietal lobe lies within the border of the central, parieto-occipital, and lateral sulci. This lobe contains the major area for somatic sensory input, located along the post-central gyrus. That Brodmann area 3, 1, and 2. I'm not sure why they just didn't say 1, 2, 3, but I guess they wanted to be different. Which is adjacent to the primary motor areas in the precentral gyrus. Communication between the motor and sensory areas is provided by association fibers. Much of this region is involved in the sensory association. The next figure here shows the distribution of function associated with both the primary motor area and the primary sensory area of the cerebral cortex. The occipital lobe lies caudal to the parieto-occipital sulci and superior to the cerebellum. The primary visual cortex, Brodmann area 17, is located in the region and receives input from the retinas. Much of the remainder of this lobe is involved in visual association, so Brodmann 18 and 19. The temporal lobe lies inferior to the lateral fissure of the and is composed of the superior, middle, and inferior temporal gyri. The primary auditory cortex, Brodmann area 41, is related association area 42 lie deep within the lateral sulcus on the superior temporal gyrus. Wernicke area, Brodmann area 22, along with the adjacent portions of the parietal lobe, constitutes a sensory speech area. This area is responsible for reception and interpretation of speech, and dysfunction may result in receptive aphasia or dysphagia. The temporal lobe also is involved in memory, consolidation, and smell. The insula, the insular lobe, lies hidden in the lateral sulci between the temporal and frontal lobe of each hemisphere. The insula processes sensory and emotional information and routes the information to other areas of the brain. Lying directly beneath the longitudinal fissure, the corpus callosum, a transverse commissural fibers is a bundle of myelinated fibers that connect the two cerebral hemispheres. The corpuscle callosum conveys contralateral projections of axons and is essential in the coordination between activities between the hemispheres. Deep inside the cerebrum are numerous white matter tracts and gray matter nuclei, and the major cerebral nuclei of the basal ganglia system. The basal ganglia system is a group of nuclei that include the caudate nucleus, putamen, and globus pallidus. The putamen and globus pallidus together are called the lentiform nuclei. The caudate nucleus and putamen together are called the striatum. The other structures in the basal ganglia include the substantia nigra, the nucleus accumbens, and subthalamic nucleus. The nuclei of the basal ganglia are important for coordination of voluntary movement and cognitive and emotional function. So here we're taking a look at the basal ganglia. The internal capsule is a white matter tract in which the afferent, your sensory, and efferent, motor pathways are conveyed to and from the cerebral cortex by passing through the center of the cerebral hemisphere and between the caudate and lentiform nuclei. The basal ganglia and their direct and indirect interconnections with the thalamus, premotor cortex, red nucleus, reticular formation, and spinal cord are part of the extrapyramidal system. The extrapyramidal system is a part of the motor control system that causes involuntary reflexes and has a stabilizing effect on the motor control. The limbic system is a group of interconnected structures located between the telencephalon and diencephalon and surrounding the corpus callosum. It is composed of the amygdala, hippocampus, fornix, hypothalamus, and related autonomic nuclei. The limbic system mediates emotion and long-term memory through a connection in the prefrontal cortex. Its principal effects are involved in the primitive behavioral responses, visceral reactions to emotion, motivation, mood, feeding behavior, biological rhythm, and the sensory of smell. Diencephalon. The diencephalon, or interbrain, surrounded by the cerebrum and sitting on top of the brainstem, has four divisions: your epithalamus, thalamus, hypothalamus, and subthalamus. The epithalamus forms the roof of the third ventricle and composes the most superior portion of the diencephalon. The diencephalon controls vital functions and visceral activities and is closely associated with those of the limbic system. The largest component of the diencephalon is the thalamus. It borders and surrounds the third ventricle and is a major integrating center for afferent, your sensory impulses, to the cerebral cortex. Various sensations are perceived at this level, but cortical processing is required for interpretation. The thalamus also serves as a relay center for information from the basal ganglia and cerebellum to the appropriate motor areas. The hypothalamus forms the ventral part of the diencephalon and functions to maintain a constant internal environment and implement behavioral pattern. Integrative centers control autonomic nervous systems and functions, regulate body temperature and endocrine function, and adjust emotional expression. The hypothalamus exerts its influence through the endocrine system as well as the neural pathways. The subthalamus flanks the hypothalamus laterally and serves as an important basal ganglia center for motor activity. Again, these boxes, please pause, read them over just so that you know them. The midbrain, the mesencephalon. The midbrain. Losing my spot here. Connects the forebrain with the hindbrain and is composed of the tectum, the tegmentum, and cerebral peduncle. The tectum includes the two pairs of superior colliculi and two pairs of inferior colliculi. The superior colliculi are involved with voluntary and involuntary visual motor movements. The inferior colliculi accomplish similar motor activities but involve movement affecting the auditory system. The inferior colliculus also is a major relay center along the auditory pathway. The tegmentum is composed of the red nucleus and substantia nigra. The red nucleus receives ascending sensory information from the cerebellum and projects a minor motor pathway, the rubrospinal tract, to the cervical spinal cord. The substantia nigra synthesizes dopamine, dysfunction of dopaminergic neurons, and the substantia nigra is associated with Parkinson's disease and schizophrenia. The cerebral peduncles of the anterior midbrain are made up of efferent fibers of the corticospinal, corticobulbar, and corticopontocerebellar tracts. Other notable structures of the region are the nuclei of the third and fourth cranial nerves, the cerebral aqueduct, the aqueduct of Sylvius, which carries cerebrospinal fluid between the third and fourth ventricles, also traverses this structure. Obstruction of this aqueduct is a common cause for hydrocephalus. Please keep that in mind. The hindbrain, your metencephalon. The major structures of the metencephalon are the cerebellum and pons. The cerebellum is composed of two lobes of gray and white matter, and its cortical surface is convoluted, like the surface of the cerebrum. The lobes are divided by a central fissure and connected by a midline structure, the vermis. The cerebellum is responsible for reflexive and voluntary fine-tuning of motor control and for maintaining balance and posture through extensive neural connections with the medulla and with the midbrain. The two hemispheres are connected to the pons by the middle cerebellar peduncles. These connections allow extensive sampling of visual, vestibular, and proprioceptive data from the region of the CNS and periphery. The cerebellum has ipsilateral control of the body, in contrast to the cerebral cortex, which has contralateral, or opposite side control of the body. The pons, that bridge, is easily recognized by its bulging appearance below the midbrain and above the medulla. Primarily, it transmits information from the cerebellum to the brainstem, between the two cerebellar hemispheres. The nuclei of the fifth through eighth cranial nerve are located all in the pons. Myelencephalon, the myelencephalon, the medulla oblongata, forms the lowest portion of the brainstem. Reflex activities such as your heart rate, respiration, blood pressure, coughing, sneezing, swallowing, vomiting are all controlled in this area. The nuclei of cranial nerves 9 through 12 are located in this region. A major portion of the descending motor pathway crosses to the other side or decussates at the inferior medulla. These pathways, together with other areas of the decussation in the CNS, are the basis for the phenomenon of contralateral control. Your sleep-wake rhythms are also processed by neural influences from the lower brain center and are associated with complex groups of diffuse structures and functions, including the reticular activating system. That leads us to the spinal cord. The spinal cord lies within the vertebral canal and is surrounded and protected by the vertebral column. One of the primary functions of the spinal cord is to transmit long motor and sensory tracts that originate on the brain and synapse with cell bodies and gray matter of the spinal cord before exiting to the body. Additional significant functions include somatic and autonomic reflexes, motor pattern control centers, and sensory and motor modulation. The spinal cord originates in the medulla oblongata and ends at the conus medullaris, a cone-shaped structure at the level of the first and second lumbar vertebra in adults. The spinal nerves extend from the conus medullaris and form a nerve bundle called the cauda equina, a thin filament. The filum terminale extends from the conus medullaris and anchors it to the coccyx. The coverings of the spinal cord mirror those of the brain and are illustrated in the following pictures. So again, please pause, take a look at these pictures so that you know what you're looking at in the future. And again, I'll pause a little bit here long so that you can read along, but again, pause if you need to. All this is technically covered in your physiology and anatomy classes. However, if any of you are like me who have not gone to school in years and years, it's nice to look at these as a refresher. So pausing is not anything to be ashamed of by any means. Grossly, the spinal cord is divided into sections: 8 cervical, 12 thoracic, 5 lumbar, 5 sacral, and 1 coccygeal. They correspond to paired nerves. The cross-section of the spinal cord is characterized by a butterfly-shaped inner core of gray matter, surrounded by white matter tracts. The central canal extends through the spinal cord from its origin in the fourth ventricle. The gray matter of the spinal cord is divided into 3 regions and displays specific functional characteristics. These regions include the posterior horn, composed primarily of interneurons and axons from sensory neurons whose bodies line the dorsal root ganglion. At the tip of the posterior horn is the substantia gelatinosa, a structure involved in pain transmission. The lateral horn contains cell bodies involved in the ANS. The anterior horn contains the nerve cell bodies for efferent, that motor pathway, that leave the spinal cord by way of spinal nerves. The terms anterior and posterior are simply preferred by many authors for describing human spinal cord anatomy, whereas dorsal and ventral are the common zoologic terms. Again, here is a picture of the ascending and descending tract of the spinal cord. Take a look, familiarize. Oh my God, I can't speak today. I hope you know what I mean. Familiarize yourself so that you understand the terms and locations. Surrounding the gray matter is white matter that forms ascending and descending pathways called spinal tracts, and short ascending and descending integrative pathways. Your spinal tracts are named to denote their beginning and ending points. An example, the spinothalamic tract carries nerve impulses from the spinal cord to the thalamus in the diencephalon. Numerous spinal tracts are grouped into columns according to their location within the white matter. These include the anterior column, lateral column, and posterior column. Your ascending tracts are found in all columns. Descending tracts are found only in the lateral and anterior columns. The following figure identifies the location and principal activity of the major spinal tracts and the horns of the gray matter. Neural circuits in the spinal cord, when activated, display specific sets of motor responses. These reflex arcs form basic units that respond to stimuli and provide protective circuitry for motor output. Structures mandatory for the reflex arc, your monosynaptic reflex, are a receptor, an afferent sensory neuron, an efferent motor neuron, and an effector muscle or gland. A simple reflex arc, such as a knee-jerk reflex, contains only two neurons. In most cases, simple reflex arcs are modulated by interneurons that provide a link between the sensory and the motor neuron. The motor effect of reflex arcs generally occur before the event is perceived in the brain's higher centers. Much internal environment regulation is mediated by reflex activity involving the ANS. So here you see the cross-section of the spinal cord showing a simple reflex arc. Afferent pathways transmit information from peripheral receptors with an eventual termination in the cerebral or cerebellar cortex, or both. Your efferent pathways primarily relay information from the cerebrum to the brainstem or spinal cord. The upper motor neurons are completely contained within the CNS. Their primary roles are controlling fine motor movement and influencing modifying spinal reflex arcs and circuits. Generally, upper motor neurons form synapses with interneurons, which then form synapses with lower motor neurons that project into the periphery. Your lower motor neurons directly influence muscle. Their cell bodies line the gray matter of the brainstem and anterior horn of the spinal cord, but their processes extend out of the CNS into the PNS. Destruction of upper motor neurons usually results in initial paralysis, followed within days or weeks by partial recovery, whereas destruction of the lower motor neurons often leads to permanent paralysis unless peripheral damage is followed by nerve regeneration and recovery. Multiple activity, the stimulation and contraction, is regulated by nerve impulses. Your motor neurons innervate one or more muscle cells, forming a motor unit, which consists of a single motor neuron and all of the skeletal muscle fiber it stimulates. The junction between the axon of the motor neurons and the plasma membrane of the muscle cell is called the neuromuscular junction. So injury to motor neurons is discussed in chapter 18, which we will be covering in the very near future. It's one of the next chapters for me to read. Your motor pathways, the tracts. Clinically, relevant motor pathways are the corticospinal and corticobulbar pyramidal tracts and the extrapyramidal reticulospinal, vestibulospinal, and rubrospinal tracts. The corticospinal and corticobulbar tract consist of two neuron chains. The cell bodies originate in and around the pre-central gyrus, pass through the corona radiata of the cerebrum, the internal capsule, and the middle 3/5 of the basis pedunculi, pons, and pyramid, and decussate in the medulla oblongata, and form the lateral corticospinal tract of the spinal cord. The lateral corticospinal tract axons descend in the spinal cord and leave the white matter tract to synapse with specific interneurons or motor neurons in the anterior horn of the spinal cord. The lateral corticospinal tract has the same somatotopic organization as the body. The corticobulbar tract axons synapse on the motor cranial nuclei within the brainstem that control muscles of the face, head, and neck. Lower motor neurons project from the anterior horn of the spinal cord through nerves to specific muscles. The extrapyramidal tracts are involved in precise motor movement. The reticulospinal tract modulates motor movement by inhibiting and exciting spinal activity. The vestibulospinal tract arises from the vestibular nucleus in the pons and causes the extensor muscles of the body to rapidly contract, most dramatically witnessed when a person starts to fall backwards. The rubrospinal tract originates in the red nucleus, decussates, and terminates in the cervical spinal cord. It's important for muscle movement and fine muscle control in the upper extremities. Your sensory pathways. The three clinically important spinal afferent, the sensory pathways, are the posterior column, anterior spinothalamic, and lateral spinothalamic. The posterior column carries fine touch, vibration, and proprioceptive information. The posterior column is formed by a three-neuron chain. The first neuron of the chain are the primary afferent neurons. They are also the sensory neuron of the reflex arc. After entering the spinal cord, the primary neuron sends its axon ipsilaterally up the spinal cord in a specific part of the posterior column, and synapses in one of the three posterior column nuclei in the medulla oblongata. The axons of the second-order neurons cross contralaterally at the medial lemniscus and ascend and synapse with specific nucleus of the thalamus. The third-order neurons originate in the thalamus and continue the tract into the internal capsule, corona radiata, and postcentral gyrus. Again, Brodmann area 3, 1, 2. The anterior and lateral spinothalamic tracts are responsible for the vague touch and for pain and temperature perception, respectively. These modalities are referred to as the protopathic. These tracts also form a three-neuron chain. However, their primary afferent neuron synapse in the posterior horn of the spinal cord, not just at the level they enter the intervertebral foramen, but in a number of spinal segments above and below their point of entry. This is an example of divergence. The axons of the second order neurons in the posterior horn across to the contralateral side of the spinal cord in the lateral column and ascend to the same thalamic nucleus as the posterior column pathway, and continue on with the posterior column pathway to the postcentral gyrus. Again, if you're not looking at these pictures, it's hard to picture what I'm saying. So make sure you pause and take a look at those pictures so you get familiar with what I'm talking about here. Your protective structures, the cranium. The cranium vault functions to enclose and protect the brain and its associated structures. The bony cranium is composed of 8 bones: frontal, 2 parietal, 2 temporal, the ethmoid, the sphenoid, and occipital. The glia aponeurotica is a thick, fibrous band of tissue overlying the cranium between the frontal and occipital muscle. This structure affords added protection to the bony structure of the skull. The subgaleal space has venous connections with the dural sinuses, and with increased intracranial pressure, blood can be shunted to the space, thus reducing pressure in the intracranial cavity. The subgaleal space is also a common site for placement of wound drains after intracranial surgery. The floor of the cranial vault is irregular and contains many foramina, or openings, that act as exit sites for cranial nerves, blood vessels, and the spinal cord. The cranial floor is divided into 3 fossae, or depressions. The frontal lobes lie in the anterior fossa. The temporal lobes and base of the diencephalon lie in the middle fossa, or temporal fossa, and the cerebellum lies in the posterior fossa. These terms are all commonly used anatomical landmarks to describe the location of intracranial lesions. Your meninges. Surrounding the brain and spinal cord are three protective membranes. So again, your anatomy, the dura mater, the arachnoid, and the pia mater. Collectively, they are called the meninges. The dura mater, or hard mother, is composed of two layers, with the venous sinuses formed between them. The outermost layer forms the periosteum of the skull. The inner dura provides rigid membranes that support and separate various brain structures. One of these membranes, the falx cerebri, dips between the two cerebral hemispheres along the longitudinal fissure. The falx cerebri is anchored anteriorly to the base of the brain at the crista galli of the ethmoid bone. The tentorium cerebelli, a common landmark, is a membrane that separates the cerebellum from the cerebral structures above. The arachnoid mater is a spongy, web-like structure just underneath the dura mater that loosely follows the contours of the cerebral structures. So again, pause, take a look at, your basic anatomy here, just so that you familiarize yourself with all the specific terms. The subdural space lies between the dura and the arachnoid. Many small bridging veins that have little structural support traverse the subdural space. Their disruption results in a subdural hematoma. The subarachnoid space lies between the arachnoid and the pia mater and the cerebrospinal fluid. Unlike the dura mater and arachnoid, the delicate pia mater closely adheres to the surface of the brain and spinal cord. It provides support for blood vessels serving brain tissue. The choroid plexuses arise from the pia membrane and function to produce cerebrospinal fluid. The spinal cord is anchored to the vertebra by extension of the meninges called the denticulate ligaments. The meninges continue beyond the end of the spinal cord at vertebra level L1 and L2 to the lower portion of the sacrum. CSF contained within the subarachnoid space also circulates down to the large lumbar cistern, which extends from the second lumbar vertebra to the second sacral vertebra. Cisterns are expanded areas of the subarachnoid space. The cerebellomedullary cistern and the pontine cistern are two of the most important cisterns. The meninges form potential and real space important to understanding functional and pathologic mechanisms. An example is the dura mater and skull lie a potential space termed the epidural space. The arterial supply to the meninges consists of blood vessels that lie within grooves in the skull. A skull fracture can sever one of the vessels and produce an epidural hematoma. So now we're talking about cerebrospinal fluid and the ventricular system. Cerebrospinal fluid, or CSF, is clear, colorless fluid similar to blood plasma and interstitial fluid. The intracranial and spinal cord structures float in CSF and they are thereby partially protected from jolts and blows. The buoyant property of CSF also prevent the brain from tugging on meninges, nerve roots, and blood vessels. There's anywhere between 125 to about 150 ml of CSF circulating within the ventricles and subarachnoid space at any given time. Roughly 600 ml of CSF is produced daily. All right, so if you're now looking at the composition of CSF, again, pause, look at some numbers. It doesn't hurt to know these things for the future. The choroid plexus is in the lateral third and fourth ventricle produce major portion of CSF. These plexuses are characterized by a rich network of blood vessels supplied by the pia mater that lie close to the ependymal cells of the ventricles. The tight junction of the choroid blood vessels provide limiting barrier between the CSF and the blood that functions similarly to the blood-brain barrier. The BBB. The CSF exerts pressure within the brain and spinal cord. When a person is supine, your CSF pressure is approximately 80 to 180 mm of water pressure, but can double when the person moves to an upright position. CSF flow results from the pressure gradient between the arterial system and the CSF-filled cavities, beginning in the lateral ventricles. The CSF flows through the interventricular foramen into the third ventricle and then passes through the cerebral aqueduct into the fourth ventricle. From the fourth ventricle, CSF may pass through either the paired lateral apertures or the median aperture before communicating with the subarachnoid spaces of the brain and spinal cord. CSF is produced continually but does not accumulate. Instead, it is reabsorbed into the venous circulation through a pressure gradient between the arachnoid villi and the cerebral venous sinuses. The arachnoid villi protrude from the arachnoid space through the dura mater and lie within the blood flow of the venous sinuses. The villi function as a one-way valve directing CSF outflow into the blood, but preventing blood flow into the subarachnoid space. Thus, CSF is formed from the blood, and after circulating through the CNS, it returns to the blood. The vertebral column. The vertebral column is composed of 33 vertebrae, 7 cervical, 12 thoracic, 5 lumbar, 5 fused sacral, and 4 fused coccygeal. Between each vertebra is an intervertebral disc. At the center of the intervertebral disc is the nucleus pulposus, a pulpy mass of elastic fibers. The intervertebral disc functions to absorb shocks, preventing damage to the vertebra. The intervertebral disc is also a common source of back problems. If too much stress is applied to the vertebral column, the disc contents may rupture and protrude into the spinal canal, causing compression of the spinal cord or nerve roots. The discs also can degenerate. Again, please pause, take a look, familiarize yourself. I'll kind of stop and pause a little bit passively, but again, pause. It does not hurt to know your anatomy. Now we're talking about the blood supply of the central nervous system. Your blood supply to the brain. The brain receives roughly 20% of cardiac output, or 800 to 1,000 ml of blood flow per minute. Carbon dioxide serves as a primary regulator for blood flow within the CNS. It is a potent vasodilator in the CNS, and its effects ensure an adequate blood supply. The brain derives its arterial supply from two systems, the internal carotid arteries and the vertebral arteries. The internal carotid arteries supply a proportionately greater amount of blood flow. They originate at the common carotid arteries, enter the cranium through the base of the skull, and pass through the cavernous sinus. After entering the skull, these arteries divide into the anterior and middle cerebral arteries. The vertebral arteries originate at the subclavian arteries and pass through the transverse foramina of the cervical vertebra, entering the cranium through the foramen magnum. They join at the junction of the pons and medulla oblongata to form the basilar artery. The basilar artery divides at the level of the midbrain to form paired posterior cerebral arteries. So again, here is our major arteries of the head and neck. Pause. And here are the arteries at the base of the brain. Again, please pause, take a look at this. Three major paired arteries perfuse the cerebellum and brainstem: the posterior inferior cerebellar artery, the anterior inferior cerebellar artery, and the superior cerebellar artery. They originate from the basilar artery. The basilar artery also gives rise to the small pontine arteries. The large arteries on the surface of the brain and their branches are called the superficial arteries or conducting arteries. Small branches that project into the brain are termed projecting arteries or nutrient arteries. The circle of Willis provides an alternative route for blood flow when one of the contributing arteries is obstructed. The circle of Willis is formed by the posterior cerebral arteries, posterior communicating arteries, internal carotid arteries, anterior cerebral arteries, and anterior communicating arteries. The anterior cerebral, middle cerebral, and posterior cerebral arteries leave the arterial circle and extend to various brain structures. Again, the next table and figure will illustrate structures, served, functional relationships, and pathologic considerations related to occlusion. So this is again a good spot to hit pause so you have an understanding of why, for instance, in the frontal lobe, your parietal lobe, your temporal lobe might be related to aphasia. Cerebral venous drainage does not parallel its arterial supply, whereas the venous drainage of the brainstem and cerebellum does parallel the arterial supply of the structure. The cerebral veins are classified as superficial veins and deep cerebral veins. The veins drain into venous plexuses and dural sinuses and eventually join the internal jugular vein at the base of the skull. Adequacy of venous outflow can have a significant effect on intracranial pressure. An example of this is in individuals with head injury. Turning or letting the head fall to the side partially occludes venous return and can increase intracranial pressure because of a decreased flow through the jugular veins. So here's the veins of the head and the neck. So here we jump now to the blood-brain barrier. That BBB describes a cellular structure that selectively inhibit certain potentially harmful substances in the blood from entering the interstitial space of the brain or CSF, allowing neurons to function normally. Your endothelial cells in the brain capillaries with their intracellular tight junction are the site of the BBB. Supporting cells of their functional interactions include astrocytes, pericytes, and microglia. The exact nature of this mechanism is controversial, but permeability is high for water, carbon dioxide, oxygen, and most lipid-soluble substances, including alcohol, moderate for electrolytes such as sodium, chloride, and potassium, and almost totally impermeable to plasma proteins and most non-lipid soluble large organelle organic molecules. This has substantial implications for drug therapy because certain types of antibiotics and chemotherapeutic drugs show a greater propensity than others for crossing the blood-brain barrier. Dysfunction or increased permeability of the BBB, as occurs in hypoxia, can contribute to neuroinflammation, cerebral edema, and neurodegeneration. The epithelium of the choroid plexus and the arachnoid membrane also provide barrier functions. So here we're taking a look at the blood-brain barrier. Again, take a look so you have an understanding of how it's laid out. Your blood supply to the spinal cord. The spinal cord derives its blood supply from branches off the vertebral arteries and from branches from various regions of the descending aorta. The anterior spinal arteries and the paired posterior spinal arteries branch off the vertebral artery at the base of the cranium and descend alongside the spinal cord. Arterial branches from vessels exterior to the spinal cord follow the spinal nerve through the intervertebral foramina, pass through the dura, and divide into the anterior and posterior radicular arteries. The radicular arteries eventually reconnect to the spinal arteries. Branches from the radicular and spinal arteries form plexuses, whose branches penetrate the spinal cord, supplying the deeper tissue. Venous drainage parallels the arterial supply closely and drains into the venous sinuses, located between the dura and periosteum of the vertebra. So that was all CNS. Now the PNS, or peripheral nervous system. The peripheral nervous system include the nerves outside the central nervous system. The somatic nervous system is the part of the PNS that controls voluntary muscle movement, your efferent nerves, and sensory information, your afferent nerves. The cranial and spinal nerves, including their branches and ganglia, constitute the PNS. A peripheral nerve is composed of individual axons, dendrites, with most wrapped in a myelin sheath. These individual fibers are arranged in bundles called fascicles. The coverings provide structural support, a blood supply, and interstitial compartment necessary for delivery of essential electrolytes to support nerve and impulse conduction. So, if you look at figure C, for instance, it really breaks down which portion, determines what. So, I'm going to see if I can make this even a little bigger. Grandpa size. Boom. Again, pause, I'm going to make this even bigger. Not at 20. There we go. I'm not keeping it at the size. I just want you guys to be able to read and see it. So, again, please pause, take a look at this. It's interesting in one sense. It's also important to know in another. So I will continue reading. I hope you paused and at least gave it a one over. The 31 pairs of spinal nerves derive their names from the vertebral level from which they exit. There are 8 cervical, 12 thoracic, 5 lumbar, 5 sacral pairs of spinal nerves, and 1 coccygeal. The first cervical nerve exits above the first cervical vertebra, and the rest of the spinal nerves exit below their corresponding vertebra. From the thoracic region, nerves correspond to the vertebral layer above their exit. Spinal nerves contain both sensory and motor neurons and are called mixed nerves. They arise as rootlets lateral to the anterior and posterior horn cells of the spinal cord. These two spinal nerve roots converge in the region of the intervertebral foramen to form the spinal nerve trunk. Shortly after converging, the spinal nerve divides into anterior and posterior rami, or branches. The anterior branch initially forms plexuses, which then branch into the peripheral nerves. Instead of forming plexuses, the thoracic nerve passes through the intercostal spaces and innervates Regions of the thorax. The main spinal nerve plexus innervate the skin and underlying muscles of the limbs. The brachial plexus, for example, is formed by the last four cervical nerves, that's C5 through C8 area, and the first thoracic nerve, your T1. The brachial plexus innervates the nerves of the arm, wrist, and hand. The lumbar plexus, L2 through L4, and sacral plexus, L5 through S5, contain nerves that innervate the anterior and posterior portions of the lower body, respectively. The posterior branch of each spinal nerve, with their many processes, are distributed to a specific area of the body. Sensory signals thus arise from specific sites associated with a specific spinal cord segment. Specific areas of cutaneous innervation at these spinal cord segments are called dermatomes. The dermatomes of various spinal nerves are distributed in a fairly regular pattern, although adjacent regions between dermatomes can be innervated by more than one spinal nerve. Like spinal nerves, cranial nerves are categorized as peripheral nerves. Most of these are mixed nerves, although some are purely sensory or purely motor. An example is your cranial nerves carry only special sensory information like vision, hearing, and taste. Your cranial nerves project two nuclei in the brain and brainstem. The following figures illustrates their locations, and the following table describes their structure and functional characteristics. Things we definitely need to know. Your cranial nerves. 1. Your olfactory. Your optic. Your ocular motor. Pause, pause, pause. Write these down, make flashcards, memorize them somehow, especially their origin, their function, and how we test. These are things that you all need to remember for your advanced health assessment courses. And if you miss any of them during your advanced health assessment courses, they will ding you because you didn't hit every cranial nerve. So please pause. I know I'm clicking through this kind of quickly. Pause, write them down, make a flashcard. Your autonomic nervous system, your ANS, regulates the involuntary function of the internal organs. The structure and function of the ANS are complex and still not well understood. Components of the ANS are located in both the CNS and PNS. However, the ANS is considered part of the efferent division of the PNS, even though visceral afferent neurons are an important part of this system. Many neurons of the ANS travel in the spinal nerves and in certain cranial nerves. The widespread activity of the system indicates that its components are distributed over the entire body. The peripheral autonomic nerves carry mainly efferent fibers. The motor component of the ANS is a two-neuron system consisting of the preganglionic neurons and postganglionic neurons. This arrangement contrasts with the somatic nervous system, in which a single motor neuron travels from the CNS to the innervated structure. Your visceral afferent neurons have their cell bodies and some sensory and cranial ganglia and their fiber processes traveling in the peripheral nerves. The CNS has autonomic areas in the intermediolateral horns of the spinal cord, the cardiovascular and respiratory centers in the reticular formation, and both sympathetic and parasympathetic areas in the hypothalamus. The CNS always interconnect all these areas. The ANS coordinates and maintains a steady state among visceral organs, such as the regulation of cardiac muscle, smooth muscle, and the glands of the body. This system is considered an involuntary system because one generally cannot will these functions to happen. The ANS is separated structurally and functionally into two divisions, the sympathetic nervous system and the parasympathetic nervous system. All right, so uh, I'm going to leave this picture up for just a moment so you can take a look at it. Again, pause, 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 pause. Here it's showing you the sympathetic and parasympathetic division of the autonomic nervous system. The anatomy of the sympathetic nervous system. The sympathetic nervous system mobilizes energy stores in times of need. The sympathetic division receives its innervation from cell bodies located from the first thoracic through the second lumbar, so T1 to L2, regions of the spinal cord, and is therefore called the thoracolumbar division. Oh, sorry, thoracolumbar division. The preganglionic axon of the sympathetic division forms synapses shortly after leaving the spinal cord in the sympathetic ganglia. Your preganglionic axons travel one of the several different pathways, directly synapsing with postganglionic neurons in the sympathetic chain ganglion at their level, or two, traveling up or down the sympathetic chain ganglion before forming synapses with the higher or lower postganglionic neuron, or 3, passing through the sympathetic chain ganglion postganglionic neurons with collateral ganglia. Some preganglionic axons form pathways called splanchnic nerves, which lead to collateral ganglia on the front of the aorta. The collateral ganglia are named according to the branches of the aorta nearest them, namely the celiac, superior mesenteric, and inferior mesenteric. These postganglionic neurons leave the collateral ganglia and innervate the viscera below the diaphragm. Your preganglionic sympathetic neurons that innervate the adrenal medulla also travel in the splanchnic nerves and do not synapse before reaching the gland. The secretory cells in the adrenal medulla are considered modified postganglionic neurons because preganglionic sympathetic fibers are all myelinated, travel to the adrenal medulla is quick, and innervation causes the rapid release of epinephrine and norepinephrine, which mediate the fight or flight response. That was discussed in chapter 11. Anatomy of the parasympathetic nervous system. The parasympathetic nervous system functions to conserve and restore energy. The nerve cell bodies of this division are located in the cranial nerve nuclei and in the sacral region of the spinal cord, and therefore constitute the craniosacral division. Unlike the sympathetic division, the preganglionic fibers in the parasympathetic division travel to ganglia close to the organs they innervate before forming synapses. With the relatively short postganglionic neurons, parasympathetic nerves arise from nuclei in the brainstem, travel to the viscera of the head, thorax, and abdomen within cranial nerves, including the oculomotor, facial, glossopharyngeal, and vagus nerve. The preganglionic parasympathetic nerves that originate from the sacral region of the spinal cord run either separately or together with spinal nerves. the preganglionic axon join to form the pelvic nerve, which innervates the viscera of the pelvic cavity. These preganglionic axons synapse with postganglionic neurons in terminal ganglia located close to the organs they innervate. So you have neurotransmitters and neuroreceptors, sympathetic preganglionic fibers and parasympathetic preganglionic and postganglionic fibers release acetylcholine, the same neurotransmitter released by somatic efferent neurons. These fibers are characterized by their cholinergic transmission. Most postganglionic sympathetic fibers release norepinephrine and thus are considered to function by adrenergic transmission. It's that adrenaline. The few postganglionic sympathetic fibers, such as those that innervate the sweat glands, however, release acetylcholine. So, here we have the autonomic nervous system and the type of neurotransmitters secreted by the preganglionic and postganglionic fibers. Make sure you take a look at that. The action of catecholamines, that's your epinephrine, norepinephrine, and dopamine, varies with the type of neuroreceptor stimulated. It should be remembered that catecholamines also are released by the adrenal medulla that physiologically and biochemically resembles the sympathetic nervous system. Two types of adrenergic receptors exist, again, alphas and betas. Cells of the effector organ may have only one or both types of adrenergic receptors. The alpha adrenergic receptors have been further subdivided according to their action produced. The alpha-1 adrenergic activity is mostly associated with the excitation or stimulation. Alpha-2 adrenergic activity is associated with relaxation or inhibition. Most of the alpha adrenergic receptors on effector organs belong to the alpha-1 adrenergic class. So, your beta adrenergic receptors are classified into beta-1 adrenergic receptors, which facilitate increased heart rate and contractility and cause the release of renin from the kidneys, and your beta-2 adrenergic receptors, which facilitate all the remaining effects attributed to beta adrenergic receptors. Your norepinephrine stimulates all alpha adrenergic and beta-1 adrenergic receptors, and only certain beta-2 adrenergic receptors. The most significant response from norepinephrine, however, is the stimulation of alpha-1 adrenergic receptors that causes vasoconstriction, which is why we use it in the ICU. Epinephrine strongly stimulates all four types of receptors and induces a general vasodilation because of its predominance of beta adrenergic receptors in muscle vasculature. So here, there's this table, actions of autonomic nervous system neuroreceptors. Hit pause, it is important. I wouldn't say necessarily make flashcards for each and every single one of them, but you can merge them together so you have an idea of, hey, this is stuff you need to know. And then I'm going to let it pause over here again, or go ahead and hit pause so that you see it. Again, there is a lot. The table doesn't stop, so actually there is an end. Your dopamine. Dopamine is a precursor of norepinephrine and epinephrine and is a brain neurotransmitter synthesized in the substantia nigra and the ventral tegmental areas of the brain. There's 5 types of dopamine receptors distributed throughout the brain. and they're labeled D1 through D5. They have varying functions, including pleasure, motivation, cognition, attention, memory, learning, that fine motor control, your sleep regulation, hormonal regulation, sympathetic regulation, penile erection, and immune function. Functions of the autonomic nervous system. Many body organs are innervated by the sympathetic and parasympathetic nervous system. These two divisions frequently cause opposite responses. For example, your sympathetic stimulation of the GI tract causes a decreased peristalsis, whereas parasympathetic stimulation of the GI tract increases peristalsis. In general, sympathetic stimulation promotes response that are concerned with the protection of the individual. For example, sympathetic activity increases blood glucose levels and temperature and raises blood pressure. In emergency situations, a generalized and widespread discharge of sympathetic system occurs and is known as the fight or flight reflex, or the acute stress response. This is accomplished by an increased firing frequency of the sympathetic fibers and by activation of the sympathetic fibers normally silent. Regulation of vasomotor tone is considered the most important function of the sympathetic nervous system. So here we're going to take a look at the sympathetic activation system. Again, stop, pause, take a look at this. It doesn't hurt to know how your body is working in this sense. And if you can kind of trace it in your own body or get the effects down in that sense, when you see questions in regards to it, you can say, hey, your sympathetic activation, that's that adrenal medulla activation that releases epinephrine and norepinephrine, that increases the strength of the contraction of the heart. So again, that pause button is our friend. Increased parasympathetic activity promotes rest and tranquility and is characterized by reduced heart rate and enhanced visceral function, leading to digestion. Stimulation of the vagus nerve in the GI tract increases peristalsis and secretion, as well as relaxation of the sphincter. Activation of the parasympathetic fibers traveling within the cranial nerves 3, 8, and 9 causes pupillary constriction, tear secretion, and increased salivary secretion. The stimulation of the sacral division of the parasympathetic system contracts the urinary bladder and facilitates the process of genital erection. The parasympathetic system lacks the generalized and widespread response of the sympathetic system. Your specific parasympathetic or parasympathetic fibers are activated to regulate particular functions. Although the action of the parasympathetic and sympathetic systems are usually antagonistic, there are exceptions. Changes in the shape of the lens for near vision require only ocular motor parasympathetic activity. Most of the blood vessels involved in the control of blood pressure are innervated by sympathetic nerves. Peripheral vascular resistance is increased and decreased by the relative activity of the sympathetic division without a counteracting parasympathetic component. To decrease blood pressure, therefore, it is important to block the continuous tonic discharge of the sympathetic system than to promote parasympathetic activity. That leads us to aging and the nervous system. The CNS mechanism involved in aging processes are extremely complex, and many questions concerning the neurologic effect of aging have yet to be answered. Some of the identified Mechanisms associated with aging are pathologic, but the distinction between these mechanisms and those that are part of the normal aging process remains somewhat ambiguous. The following box contains information on the structural, cellular, cerebrovascular, and functioning changes that occur in the nervous system with aging. So again, please pause, read these over. It can't hurt to know these things, especially if you work in a geriatric facility or unit. So what kind of tests of the nervous system and structure and function? Your skull and spine, your Roentgenograms, your X-ray films of the skull or spine from multiple angles or views are used primarily to localize bony defects, bone density, erosion, or calcified structures. The pineal gland in older people become calcified and is useful as an internal brain landmark. Probably the most commonly used radiologic study are X-ray films. CAT scans. Computer tomography or CAT scans create two-dimensional reconstructions from multiple radiologic images. Using computer-assisted analysis, it is capable of demonstrating fine distinctions in shape, size, and density of a variety of tissues based on different absorption of X-rays. CT imaging is a non-invasive procedure. We're all familiar with it. A variety of contrast media is commonly used in conjunction with the procedure to aid in enhanced delineation of selected structures. Your spiral or helical CT uses a multi-detector scanner that's rotating to provide several axial images. And again, we can use our helical CT angiogram that uses contrast media to detect aneurysms, ruptures, and strokes. We can use our MRIs. Again, we're all familiar with MRIs, X-rays, CAT scans, and how we use them to provide diagnostic results. The good things about with the MRI is that they can be completed at the same time as a regular or an MRI can be done at the same time as your MRI, which can analyze the chemical composition of protons or phosphorus-based molecules. Your functioning MRI detects changes in blood oxygen, and your diffusion tensor imaging is an MRI technique that quantifies the size and direction of white matter. So there are different types of MRI scans that we can test to assess brain function and how strokes may affect that. Your angiography uses special imaging techniques, for instance, to visualize blood vessels. Your PET scan uses a CT image to detect emission of positive electrons from trace amounts of radioactive substances injected into the bloodstream. So we can use that to assess for glucose and oxygen uptake, cerebral blood flow, neural and neurotransmitter function, and the effects of certain drugs. Your single photon emission CT uses radio tracers that have longer half-lives than PET scans, but also can test a couple other things. Again, I think for the most of us, these are all straightforward tests that we're all very familiar with at this point. Your cerebral angiogram, again, it looks at the overall blood flow of the brain. Ultrasounds and your echoencephalography. Oof, butchering this stuff. Electroencephalopathy, your EEG. Might be some people are not familiar with an EEG. They are the electrical impulses that arrive from the cortical surface of the brain, and they're usually used to detect seizure activity. And then your magnetoencephalopathygram is a specialized imaging technique used to measure magnetic fields induced by electrical activity in the brain. It's usually used in correlation with an EEG. Your evoked potentials are methods of detecting electrical brain activity that result from a stimulus, either primarily auditory, visual, or peripheral. Your cerebrospinal fluid analysis. Your CSF generally is obtained from the lumbar or cisternal subarachnoid space by means of a hollow needle that allows passive flow. The lumbar puncture is performed most often between L3 and L4 interspace. The cisternal puncture is performed by the insertion of a needle through the cerebellomedullary cistern, using an approach from the back of the neck to the region of the foramen magnum. Your CSF pressure is commonly measured during this procedure, and CSF can be analyzed also for gross characteristics of constituents, your color, blood cells, electrolytes, and proteins. And we can culture it for microorganisms. And this kind of concludes this chapter. I know I kind of skipped over the testing portion of it, but I believe most of us are registered nurses or physician assistants or are in med school and have heard of CAT scans, MRIs, and the variety of ways that we can utilize them. Again, here are ways that we can, or here are measurements rather, of our CSF. So please make some flashcards. Learn your ratios, learn your, what is our norm, what is high, what is low. It does not hurt to know these things. Again, hit pause. I'm going to go back up a little bit because I chatted kind of over it as I was going down. But if here are your parameters, your normal, your abnormal, and what could be the cause, it can't hurt to know that. Again, that pause button is a beautiful thing. But this is where I will call it for a close. And I'll see you guys with chapter 16 coming next. And that is your pain, your temperature regulation, sleep, and sensory function. So, I thank you all for watching, for being part of this lovely community, and I'll see you next time.